Okay, so what we have here in order to understand what happened in the GI system, you can see we have two main parts. We have antrum and we have fundus for the GI system. When you speak about the GI system, okay, so we have the fundus and then we have the antrum here. And in the antrum, we have two type of cells. As you can see it, we have G cells and then we have D cells. And we have G cells and D cells. So the G cells, as you can see, responsible for formation of gastrin. And the D cells responsible for formation and release of somatostatin. So in the antrum here, responsible for two type of mediator, somatostatin from D cell, gastrin released from the G cells. Somatostatin under the effect of acid will be released, and this gastrin will be released under the effect of dietary food. Okay, so what we have here, we can look on the some maybe a process there is very important. Okay, so what we have here, somatostatin, the green circle here, this is a somatostatin. The somatostatin released from the D cells. Okay, somatostatin, when the somatostatin released from the D cells, what happened here? Somatostatin will affect the gastrin, these G cells, and inhibit release of gastrin. Somatostatin considered like inhibitory hormone. So somatostatin is hormone. So when somatostatin released from these cells, this will lead to the inhibitory effect on the gastrin, inhibit secretion of gastrin, somatostatin. Okay, if you don't have this effect here between the GI system, only we have stimulation of G cells. G cells will produce gastrin. Gastrin is a type of hormone. Gastrin released from the G cells enter the blood. You can see it in the blood here, gastrin moving on the blood. Okay, that's great. And until arrive to the fundus. Okay, this place here arrive to the fundus. You can see the gastrin, and then gastrin bind to the receptor. This receptor called gastrin CCKR, calcitonin receptor. So gastrin bind to the calcitonin receptor. This will lead to the stimulation of histamine fascicles. Okay, the histamine fascicles and enhanced release of histamine. And this cells called ECL cells. And ECL stands for enterochromaffin cells. So we can see here we have four main cells in the beginning. Number one, okay, we have four main cells there. Number one, we have G cells produce gastrin. We have D cells produce somatostatin. Enterochromaffin cells produce histamine. And then we have parietal cells. This is produce the acid. You can see the proton here. So we have four main cells involved in the peptic ulcer disease. Number one, G cells, gastrin. D cells, somatostatin. Enterochromaffin produce histamine, and parietal cells produce the proton. Okay, so you can see here, as we discussed, the gastrin release from the G cells enter the bloodstream and going to the enterochromaffin cells to induce and release of histamine. Okay, not only gastrin here affect the histamine. They said we have another factors may release histamine. This factor is called acetylcholine. Acetylcholine is like released from the neurons. We have some neurons release acetylcholines. Okay, this coming from autonomic nervous system. Okay, autonomic nervous system neuron release acetylcholine in the enterochromaffin cells. We have receptor related to the acetylcholine. This receptor called muscarinic receptor, and we have a specific type of muscarinic receptor called M3 receptor. So M3 receptor will be activated. So acetylcholine will activate M3 receptor in the enterochromaffin cells, and this will lead to activate release of histamine. So you can see here we have muscarinic receptor activate histamine release. We have gastrin activate histamine release. We have several factors maybe lead to the activation and release of histamine from the cells. Okay, so now the second step, you can see it here, release of histamine. This is the second step. So releasing of histamine. When the histamine released, in the between the cells, histamine will bind to the histamine receptor. What type of histamine receptor we have in the parietal cells? H1 or H2? What do you think? So if you said H2, you are correct. So H2 receptor will be available in the parietal cells. Okay, H2 receptor. Okay, this is number one. Again, what happened there? We have M3 receptor muscarinic, and we have, they said, okay, you can see the acetylcholine. Also, acetylcholine activate and stimulate M3 receptor in the parietal cells. And okay, so acetylcholine stimulate muscarinic receptor in the parietal cells. In the parietal cells also we have a gastrin receptor. So gastrin receptor here will be activated. So from these cells here, the parietal cells is the most important. Our target for treatment will be parietal cells. So this is our target, parietal cells. How many receptors do you have in the parietal cells? We have 4 main receptors, as you can see here. We have muscarinic receptor, M3 receptor. We have histaminic receptor, H2 receptor. We have gastrin receptor. And receptor number 4, we have prostaglandin receptor. So we have 4 main receptors available in the parietal cells. I will repeat it. Muscarinic receptor, histaminic receptor, prostaglandin receptor, and gastrin receptor. Okay, all these receptors, when it's activated or inhibited, will interfere with the proton pump inhibitor, with something called proton pump. Another name, proton pump. So another name could be hydrogen potassium ATPase. So you can see here we have several steps to release acid from the GI system. So we need you to remember from this graph here what type of cell will be involved, and what is the effect of these receptors on the cells. The next slide will discuss the effect of these receptors, 4 receptors, the effect of these 4 receptors on the production of acid.